A comprehensive new genetic and statistical study from researchers at the University of Kansas reveals two groups of scrub jays, one in Mexico and one in Texas, deserve status as independent species. The paper, appearing in Systematic Biology, also uses genomic data to sketch a natural history of scrub jays, showing how geographic changes over millennia split up and reconnected groups of the birds, swaying the flow of genes between them. Scrub jays are cousins to blue jays, said lead author Devon DeRaad, doctoral student at the Biodiversity Institute and Natural History Museum at Kew. In the western U.S., they're common backyard birds for people, similar to blue jays that take peanuts from your feeder. They're exciting birds because they're charismatic. They'll come to your feeder and interact. They're jays, so they're extremely smart, and they have some of the best spatial memory of any birds. They can remember thousands of places that they've cached seeds and things like that. According to the study, genetic sequencing at Kew of scrub jays collected decades ago reveals evidence of two distinct new species in Aphylocoma, the scrub jays genus, Asumichrasti, a phenotypically, behaviora. We weren't sure how different it was, he said. It's a bit of an isolated population on the Edwards Plateau in Texas, a restricted, small region of Texas. It was named as a subspecies so there are small phenotypic differences of size and the color, but it doesn't look too different from the rest. We did as many analyses as we could think of to rigorously prove ourselves wrong, but everything indicates this population in Texas is totally distinct and evolving on its own isolated trajectory. DeRaad first became interested in the biodiversity of scrub jays upon learning the variety of specimens. My goal was to use genetic methods for my Ph.D. and sequence DNA from these birds to figure out how many species there actually are. As a graduate student at Kew, DeRaad accessed genetic samples of scrub jays collected decades ago by co-author A. He collected the specimens for his dissertation between around 1989 and 1992, and this was at the very beginning of people taking tissues from specimens, DeRaad said. It used to be you'd collect the birds and stuff them with cotton to make museum specimens. Skin and feathers are very stable and will be fine in a drawer untreated if they're kept away from light and bugs. But before people were doing DNA analysis, they didn't know to keep the tissue. So, it's really cool that Town published analyses on these birds about 30 years ago with some of the earliest DNA methods, and were able to use literally the same tissues for this study. In addition to Peterson, DeRaad's co-authors were Robert Moyle, senior curator at Kew's Biodiversity Institute. In addition to determining the species status of Asumichrasti and A. texana, DeRaad and his colleagues sought to shed light on the natural history of scrub jays and draw a more accurate evolutionary tree for the birds. For instance, the team found two scrub jay species living on a genetic tree with snarled branches, showing complex signatures of both ancient and modern gene flow between the non-sister California scrub jay, Aphylocoma californica, and Woodhouse's scrub jay, Aphylocoma woodhousei, that result in D. We're coming to a more nuanced understanding of what a species is, allowing for the biological reality that evolution is more complicated than even people who study it could possibly imagine, DeRaad said. Trying to reconstruct that isn't simple, DeRaad said. We don't have a time machine, so we'll never accurately reconstruct it all minute by minute. but by being able to get genomic data from thousands of pieces of DNA from all throughout the genome, we have much more statistical power to try to reconstruct those events than we did even just 10 to 15 y. University of Kansas. 2022, July 13. Genomic research supports recognizing new scrub jay species in Texas and Mexico. Science Daily. Retrieved July 13, 2022 from ift.tt/UNPOFT University of Kansas. Thanks for watching. Please subscribe my channel for more videos.